let's get started. Let's get started. Okay, so not much business today. Don't forget to bring Scantons. Those who still haven't brought me their big ones for the final, please bring it to recitation. Don't forget there's a quiz this weekend. There's a quiz every weekend. I'll remind you, but hopefully you guys are going to get autopilot about, oh, there's a quiz I should be doing. Okay, so let's take 5 minutes to write, okay? Okay. Proteins move between compartments in transport vesicles. Explain the basic mechanism behind vesicular transport. Make sure to include cargo packaging, vesicle formation and release, and fusion with space, acceptor compartment. What would be the most likely outcome if there were a deleterious mutation? Deleterious mutation of that in the clathrin binding domain of adaptins. Okay, so take your 5 minutes, look through your notes, and we'll discuss. There's just no way to win with this thing. Okay, guys. That's 5 minutes. So, where's Tasha? Tasha? She's going to hear that later. Okay. No Tasha. Erin Phillips. No. Okay. So, let's start with the first part. Explain the mechanism behind vesicular transport and make sure you include all those things. So tell me what you were able to put together.
I said the cargo is kind of concentrated in a certain area of the cell, which forms the curvature of the vesicle. And then adaptin binds to the coat, which is the part. And dynamin pinches off the coat and releases it into the site as well. And then the snares kind of play a part. There's the vesicle snares and the target snares, which intertwine and push out all the water and fuse the vesicle into the target compartment. And then NSF unlines the snares as it's fused into the target.
Okay, that's pretty good. So what I'm hearing is concentrate the cargo. There's actually a receptor that will bind to the cargo that will help to concentrate the cargo in the ER into a certain location in the ER. And then adaptin will bind to the receptor, and then clathrin or whatever coat we're talking about, in this case, clathrin will bind to adaptin, and that will help with making the curvature of your vesicle. Dynamin will help with the pinching off of the vesicle, and then it'll move towards the next compartment, and then snares are involved in fusion. Okay, does anyone want to add to that? Just check over here. Okay. So what would be the most likely outcome if there were a deleterious mutation in clathrin binding domain, in the clathrin binding domain of adaptin? Where's Shauna? Yeah.
Wouldn't it just not form correctly?
Well, so I want more specific than that. So an obvious way to always answer a question when there's mutation is say this won't work. But I want more detail than that. What won't work properly and what will be the outcome? So let's take someone else. Let me check you off. Okay, does anyone want to take, so yes, it's not going to work properly, but what's not going to happen specifically? Yes? It won't allow the proteins to be concentrated in the vesicle, and so then they won't be transported. So the best way to approach this kind of question, of course I've gotten a paper. The best way to approach this kind of question is to draw it out, right? And so you can be like, okay, here's your ER, and here's your receptor for the cargo, and it's going to bind there. And then adaptin is going to be this thing that binds there. And then clathrin is going to bind there. And so I'm saying that the mutation is going to be here. And so the question is, will you still be able to concentrate the cargo? Well, the cargo will still be able to bind to the receptor. Will it be able to move over together to be concentrated? That's questionable. That would be one of those things where I'd have to think long and hard on how I would grade that. Because it's possible, because it's the the whole thing coming together with the clathrin that kind of brings it all together into a vesicle. And so yes, you're right, the cargo won't be incorporated into a vesicle and it won't be transported. And so you're right, it won't be transported. But when you always have to think to yourself, okay, how does this structure or process look? Where is she saying there's a mutation? What does that part do? What will that outcome be? So here, adaptin can still bind to clathrin, but it can't bind to the receptor, so there won't be any vesicle formation because the clathrin is required for that curvature. So anything that sounded like that would be right. Yes?
So then would that lead to an accumulation of cargo inside the cell then, and then it could lead to apoptosis?
So would that lead to, so a lot of people are saying cell. I want you to correct that to ER, right? So wouldn't that lead to an accumulation of protein inside the ER? Would that lead to apoptosis? Because are you taking the leap to unfolded protein response? So the only thing is these are technically properly folded proteins, so they shouldn't elicit the unfolded protein response, but they probably would elicit some sort of ER stress response. So saying apoptosis is not completely, I don't know the exact response, the outcome, but it's not completely impossible. You could think that there could be some huge amount of stress in the ER from accumulation of proteins, so that would not be wrong. Anyone else have something they want to say? Something they wrote down that was not in agreement with what we spoke about, but they feel is right? And because people kind of look at these questions in very different ways, and often are right, even though it's not exactly what I was looking for. If you're not wrong, then you're right. Right? Yes.
I'm just curious. The adaptin and clathrin and diamond, are they only working in the ER?
So this is so the question is, clathrin, adaptin, diamond, are they only working in the ER? In reality, this is not about ER transport. This is about vesicular transport. And I'm using clathrin, adaptin, diamond, DAP system as a model for vesicular transport, but really there are multiple coat proteins. And so the different coat proteins work in different compartments. So there's COP1, COP2, COP1.
Different, but they basically resemble.
Similar process, different names. Yeah. Yep.
Is it possible that when the receptor binds to adaptin, that adaptin might have like a conformational change and that allows clathrin to bind to it? So if the receptor were unable to bind to adaptin, then clathrin would be able to bind as well?
Exactly. So is it possible that adaptin is required for clathrin binding? That is the job of adaptin.
What I'm saying is that would adaptin have to bind to the receptor in order for clathrin to bind to adaptin?
Okay, so could clathrin bind to adaptin in the absence of a receptor? I kind of feel like, I'm not 100% sure if there's a conformational change that happens in that whole process, but I want to say that unless there is some sort of structural change going on, there's no reason that adaptin shouldn't be able to bind to clathrin, but it's not going to do anything, right? Because it won't be up the membrane. Okay, any other thoughts? Okay, good questions. So these are the kinds of questions to start getting used to. If you're sitting through these and you are extremely lost, you need to be seeing me often. Okay, so far, what have we done? A whole lot of fun stuff, right? Intracellular traffic, secretory pathway, the import of proteins into the ER, how integral membrane proteins. Actually, so someone came to visit me yesterday and was asking where integral membrane proteins come into the whole thing. Is this part, what part of the pathway is this? And it made me realize that it wasn't, that there was a, maybe the way I was explaining it made it sound like it was a different thing. And so what we did in my office hours is I kind of did this kind of hierarchy chart where I said, well, you've got, import. Can everyone see this? Maybe? A little bit? Okay. do this. I need to remember to bring paper. Sometimes people leave me paper here. Okay. So you'll have like import of proteins, right? And then under that, so if that's like Roman numeral 1, you can have A and B, and then you would have soluble proteins and membrane proteins. And you might ask yourself, what's soluble protein? Well, just stop and think about it for a minute. Something that's hydrophobic is not going to be soluble, right? A transmembrane domain is hydrophobic, so it won't be soluble. It's in a membrane, so it's a membrane protein. Something lacking a transmembrane domain, which is a protein that would be cytosolic, is going to be soluble because it lives in a watery environment. Does that make sense? Mm-hmm. So what you would think to yourself is, okay, there are two categories of proteins that are imported into ER. We spoke about how soluble proteins get imported, and we spoke about how membrane proteins get imported. And then the whole process kind of continues, okay? I'm hoping that clarifies things for a few people. You had a question? Oh, next time come visit me before class. Actually, I'm just going to bring the whole stack and leave it in here. Okay, thank you. Okay, anywho, I digress. Processing and modifications of proteins in the ER, protein folding, quality control, ERAD, UPR, ubiquitin proteasome system, transport from the ER to Golgi, so packaging, budding, targeting fusion, and then we kind of stopped with GTP binding proteins, right? And when I was talking about molecular switches, and that this is a theme, and we're going to see it again and again, and that GEFs activate because they'll get rid of GDP and add a GTP, and GAPs turn things off because they hydrolyze GTP to GDP, right? I was talking about that, and then we did this video. We looked at the video. We saw how whether or not you have GTP bound or GDP bound, that changes the conformation of the GTP binding protein, and that's how it's acting as a switch. If it's in one conformation, it's going to be likely to bind to one thing, and if it changes, it's going to lose a binding site. So that's a major concept. So I want everyone to just stop and think about it for a minute. Right? You might say, think about what? Think about a protein and how the job of a protein is to do something, usually through binding to other proteins. That's really how they work, right? And we talked a little bit about post-translational modifications and how they might affect binding, but you also have the situation where the GTP binding status is going to change the conformation of a protein, and that will change its structure, and that will change its ability to bind other proteins. So the example in this figure here, you're seeing this protein here, and so it might have this GTP binding site, right? And so it's got those 3 phosphates, right? And so this helix might be positioned right here when there's GTP, but when you get GTP hydrolysis and you get GDP, look, it changes. The whole thing changes, right? Do you guys see how the structure of this protein changes? Okay. And so this is something I really like to drive home because I think the concept of understanding what proteins actually do in cell biology and molecular biology is pretty huge. Because I think people come into this level of classes understanding things at the level of there are proteins and they have structure, but not really truly understanding what that means. So I want you to understand that. If you don't understand it, come see me and we can talk about it. Okay. So why am I talking about GTP binding proteins? Well, the first one we're going to talk about is RAB. And so earlier on I said, oh, RABs and snares are involved in reciprocal transport of targeting and free fusion, right? So RAB is a GTPase. It's a monomeric GTPase, so it's one protein with GTP hydrolysis activity, GTP ACE, right? And it helps determine specificity of vesicle targeting. So in this figure, you can see you've got your vesicle, and you've got your RAB that's GTP bound, so it's in one conformation, and you've also got your V snare, and here's your receptor binding to your cargo protein. This would be a soluble protein, right? And then what it would do is it binds to a RAB effector. So that's just almost like a receptor for RAB that's going to be on the destination compartment, the target compartment. And so you've got your snares here too. And you're going to get docking, so there'll be interaction, binding between RAB and the RAB effector protein. And then that brings that vesicle, the snares close together, and so that they can wind around each other and start to initiate fusion. And then you'll get GTP hydrolysis, and then that will release the RAB. So you're having binding and release. I think I've got another one here. Well, first of all, I want you to notice there are different RABs that are involved with different compartments. So there's specificity of transport. Certain RABs that are specific for ER and Golgi, certain RABs that are specific for different endosomes. And so there's specificity in transport, which kind of makes sense. If vesicles were just kind of bubbling around the cell, then there'd be no directionality. You would have ER to lysosome and Golgi to ER, and we have that anyways, but there's purpose to it, right? Because proteins are going in a certain direction for a reason. So here's another way to look at it, right? So you've got your donor compartment up here and your target compartment at the bottom. So your donor compartment could be the ER, your target compartment could be the Golgi, right? And you'll have a GEF that actually is hanging out in the membrane of your donor compartment. And remember, GEF, what it does is it will remove the GDP and replace it with GTP to turn on RAB. And then RAB GTP will bind to the vesicle, and it will go to the target compartment, bind the RAB effector. Snares will bind to each other, you get fusion, and then you'll get GTP hydrolysis, and then RAB releases and goes away. So remember, what we're really seeing here is that binding and release activity with RAB, which is regulated by conformational change, which is determined by the GTP binding status. Different RAB proteins and different effectors in different organelles. So that's that specificity, okay? Yeah. Any questions about this? Okay, so proteins, they're moving out of the ER in their vesicles, and actually the vesicles will fuse together into vesicular tubular clusters. And so it's not just an individual vesicle that will move to the Golgi. Often they'll kind of fuse to each other along the way and then fuse with the Golgi. So this is kind of showing how you're going to have your proteins that are budding, your vesicles budding out of the ER, carrying that COP2 coat. Remember, different coats for different types of transport. And then they're going to fuse together into this vesicular tubular cluster that's going to move towards the Golgi. Notice they're bound to motor proteins moving along microtubules. Remember, at some point, someone has asked about how they get to where they need to go. Motor proteins dragging. Remember we watched that movie in the beginning that was showing vesicles and attached to motor proteins walking along microtubules, so that's what's happening here.
Quick question.
Yeah.
Are the coats signal for the proteins within the ER to go to that area, or are they signal to go to a certain compartment?
So the question is, what's the purpose of the coats, right? Like to make it more broad. So they're really about making the curvature of the membrane for the vesicle formation. They are specific. So you're now wanting to know why are they different in different compartments? Is that your question? Kind of, maybe. And I'm not 100% sure why they're different. They are different. I don't know.
They go to different areas.
They go to different, so they come from different places. They go to different places.
Is the different coat telling the other compartment like this is the correct place that I need to be, or is
That's
that
RAB.
the RAB?
Yeah, so RAB is involved in the targeting and the snares also. So the combination of RAB and snares, that's your specificity and transport and targeting to the correct compartment.
Okay, so code is only for the structure.
Code is structure. I don't, I never want to say something definitively
Right.
because I'm not an expert in everything. And you have to ask yourself always, if there are different types of codes, what's the purpose, right? And so that could be an interesting thing to look into. And so here I wanted to point out that there's a difference here, right? So you've got proteins moving in two directions. So what's going on here? Why are proteins moving in two different directions? And why do they have two different kinds of coats? Well, because they're moving in two directions. But why? What's going on? Yep. Is it because of a protein chaperone? So give me more information than that. Expand. So you're talking about proteins want to go back. Is that what you're saying? Okay. So what kinds of proteins want to go back to the ER? So things might move to Golgi from the ER to Golgi, and then some things need to go back. Yeah.
So proteins that were made in the ER had to go to the Golgi for further modifications. Then they need to go back to the ER after getting those modifications from the Golgi. Will go back to the ER.
Okay, so I want you to cook that idea a little bit longer. Because you're like getting there, but you're missing a part. So I hear proteins are made in the ER, they go to the Golgi for further modifications, and then I'm hearing proteins go back to the ER. Which proteins go back to the ER for what purpose? Yes. Great, proteins that work in the ER have to go back to the ER. So not all proteins go to the Golgi and back to the ER, because then we would just be like back and forth, back and forth, back and forth. But certain proteins go. So some proteins, all proteins in the secretory pathway are made in the ER and go to the Golgi. Some go on to other places, but some work in the ER, so they need to go back. And so I think that's what I want to call you, Tamika. Am I right? Is that your name? Huh? Tiana. Is there a Tamika? I'm so sorry. Are you guys going to see a resignation? Okay. I'm sorry. I swear I don't think people think like we're anything. Tiana. I'm getting there. Yes.
So is snare one form of way that they can transport messages from one to another? This is another one that's specifically for the
No, okay, so the question is, like what's snares doing, what's this doing, are they different, are they the same, are they part of? So snares are involved in vesicle fusion. That is their job. This is just talking about the general scheme of there's this forward flow of proteins and then there's this backwards flow of proteins that need to go, resident proteins of the ER might move forward but need to go back because they belong in the ER. Snares are involved every single time a vesicle is fusing with the target memory. Okay? So you're having vesicle fusion as you're going ER to Golgi, snares are involved. You're having vesicle fusion as ER resident proteins are leaving the Golgi to go back to the ER. There's vesicle fusion there too. And those snares are involved, and RAB are involved in this. Okay, any other questions about this? Yes.
So when you're forming the tubular clusters, do they all have the same type of RABs since they're all like going together to the same exact place?
Yeah, so since you're forming vesicular tubular clusters, you can make the assumption that you would have the same RABs because they're all coming from the ER and going to the Golgi. But there would probably be different RABs in going from ER to Golgi, ER to vesicular tubular cluster, which kind of goes to Golgi, or versus an endosome that's going to come in. Okay. Anything else? We good? You look like you're confused.
The reticular tubular cluster is just the proteins what you are found from Golgi. But then why would it, if they didn't reach the Golgi, why would it go back to the ER?
No, this is not a circle. This is not a circle. So, okay, so I see we're getting confused. Vesicular tubular clusters, yes, it's just a bunch of vesicles fusing together en route to the Golgi. That's all it is. They don't turn, it's not a circle. It's not like then they come back. It's just if you think that there's a flow of proteins that's going ER Golgi outwards, there's also a flow of proteins coming back. It's not the same ones. It's only ER resident proteins that are coming back. Yes?
Are they always have to form clusters, or can they individually?
Do they always have to form clusters, or can they individually fuse? I think they can individually fuse, but I think they tend to form clusters. I think if you somehow inhibited vesicular tubular cluster formation, I'm sure it wouldn't be unable to fuse. Who else had another hand? Yep. You're making connections. Great. So the question is, on the quiz, there's something about a protein with the amino acids KDEL. Is that a protein that would go back to the ER? Yes. That's these KDEL proteins, and we're going to talk about that in just a second. Yeah. Yes.
So why would vesicle with different destinations come together?
No, so these are not vesicles. So the question is, how come vesicles with different destinations are clumping together? They're not. They're going to the same place. All these ones with COP2 are going from ER to Golgi.
Yeah, but there's the bottom one that's going back to the ER, but the rest of them are not.
Right. So proteins go where they need to go. So proteins with KDEL are going, which we're going to talk about it formally in a second, are going to bud from the Golgi and head towards the ER. Proteins that need to go to the Golgi will go to the Golgi and often form vesicular tubular clusters. Yes?
You said that if you had the right to, I guess, put engineer a signal in a protein for it to go to the ER. So if this protein is not supposed to go to the ER, would it have the ability to blot off and go to where it's supposed to? Or would it be greater?
Okay, so, hey guys, wait 1 second here. So, you're saying, so we talked about at some point, I'm remembering this conversation, how you can engineer a protein to go to the ER by addition of KDEL. And so, yes, you add KDEL to any protein, it will go return to the ER. As long as it has a signal sequence to get into the pathway. But anyway, so what's your second part of your question?
So if it's not supposed to be in the ER, would it have the ability to like blot out into a vesicle and go to where it's supposed to go? Or
Yeah. So it's not supposed to live in the ER. It's not going to have KDEL, and it won't be returned to the ER. It's going to continue that outward flow of the secretory pathway.
so you could assume that all the surfaces of the organelles would be the same if they have the ability to fuse to each other.
You lost me there. So you can assume that the surfaces of the organelles are the same because they have the ability to fuse to each other. They don't have the ability to fuse to each other. In the secretory pathway, anytime your Rab and Rab effector match up and anytime your snares match up, then they fuse to each other. Okay? Okay. Lots of questions. I hope these questions are helping everyone to understand things. Did you have a question? You guys were having a lot of conversation. It was almost like a debate going on.
Wait, I was just asking if the middle, after COP2 code in the middle of the diagram, that's
Yep, so these are your vesicular tubular clusters, and they're going to go here and fuse with the Golgi. Yep. Okay. Are we ready to move on, or do we have more questions? Okay. Good. Retrieval. Huh?
Answer the last question.
Difference between COP1 and COP2. They're different protein codes, and they're associated with the different flow of proteins from different places. So if you're going from ER to Golgi, you are COP2. If you are going from Golgi to ER, you are COP1. Okay? Yeah. Just look at this figure here. So the question is what's going on here? Difference between COP1 and COP2. COP2 is a code that originates in the ER, so it's involved in flow of proteins from the ER to the Golgi. COP1 is a protein code that's associated with the Golgi and movement from Golgi to the ER. And actually, that could be a really great question for an exam, right? I could say something like, you're trying to find out where a certain protein goes, and you notice that it has a COP1 protein code. What's its most likely destination, right? Yes.
Do the protein codes like pop off vesicles once they're formed?
Yes. Protein codes are released after the vesicle pinches off.
So like the little buds that come out of the bottom, the tubular clusters, those look like they come off before they get to the gold.
Right, yeah. So in all these right here, you see the protein coat forming where you have your vesicle forming and then it's gone. That's because it releases. Yeah.
So it doesn't have to reach the Golgi in order to.
No, yeah. Protein coat releases as soon as the vesicle buds. Yes, yes.
Where does the protein code come into play with regards to the adaptin and clathrin?
Where does a protein coat come into play with regards to adaptin? Oh, I was wishing I was still having that figure. Adaptin and clathrin. That was this one I just talked to you guys about. Clathrin is a protein coat. So let's just take a quick look at this picture I drew before. Right, so I said, here's our clathrin. Here's our adaptin. And then here's your receptor, right? Cargo receptor. Clathrin is a protein code.
So
That's one example. We talk about clathrin, COP1, COP2. There's another one. I have to think for a minute. Yeah.
So clathrin is a type of COP?
Clathrin is a protein coat called clathrin. COP1 is a protein coat called COP1. Same function in forming the curvature of the membrane and making the vesicle and concentrating the cargo. Okay? Does that make sense to you guys? Are you guys so tired of this and you just want to move on? I like to linger on topics when there's a lot of questions, because I always assume if there's a lot of questions, then a lot of people who don't want to ask those questions are going to benefit. But you're right, sometimes it's too long. Okay, so here's again, so this is good for what you just asked, right? So here's we're talking about ER resident proteins, heading back to the ER. So here, here's COP1. So this would be the Golgi, right? And so you'll have an ER protein that's in here with KDEL, and it's going to bind to a KDEL receptor, which then is going to bind to the protein coat and bud off in the ER. You can easily interchange that whole kind of process with the adaptin clathrin process, where you have a receptor, a cargo receptor that binds somehow to a protein coat. Okay? And so here what happens is the ER resident proteins that have this KDEL sequence, they're going to bind to the KDEL receptor and get concentrated in a vesicle. And then they're going to move backwards from the Golgi to the ER. And then they'll be released in the ER. And so something I want you to point out, or I want you to think about, and I'm going to point out, is there's a pH gradient as you go through the secretory pathway. In the ER, it's less acidic than the Golgi. So you're going to get increased acidity or decreased pH as you go from ER to Golgi. And if you think back to chemistry or biochemistry, or just think about it if you haven't thought about any of that stuff in a while, protein interaction is affected by pH, right? Or you can think binding of things is affected by pH, right? And so in one situation, at an acidic pH, it might be the ideal pH for KDEL to bind to the KDEL receptor. But at a neutral pH, it's not going to bind very well. So that's how these proteins are released in the ER. Okay? So I keep saying KDEL. Just understand that this is a sequence that's in a protein that lives in the ER. This is part of the protein sequence. We're talking single letter amino acid code. Understand its function. It's a very nice, I love mechanisms, and this is a very nice mechanism, so it's important to understand in great detail. So understand its function, understand how KDEL receptor knows to release the protein in the ER, and not in the Golgi or vesicular tubular cluster. Think about what if KDEL is missing. So what would happen if you have an ER resident protein with no KDEL sequence? Where would it be? Yes. Somewhere else, not in the ER. What's the end? So what's the default destination for the secretory pathway?
Outside the cell.
Outside the cell. Exactly. So if you have BIP, which is a HSP70 chaperone, oops, I think I just gave away a practice problem. Then if you don't have the KDEL, it might get secreted, right? Other mechanism for retaining, why did I write that? Other mechanism for retaining ER or Golgi resident protein. I wrote that for a reason, I don't remember what it is. Someone emailed me to remind me to let you guys know. Yes? Huh? All ER resident proteins have the KDEL sequence, yes. Yes. Okay, so here's the practice problem I just gave away. So this will test whether you understood what I was talking about. Okay, so here's our practice problem. So you're studying a protein that appears to be a member of the HSP70 family of chaperones. You believe it is an ER resident chaperone. You look at it using immunofluorescence, using a BIP antibody, which is green, and you get the following result. So you see, remember, blue is DNA, so that's your nucleus. Remember, ER and Golgi kind of come right off of the nucleus. So you're seeing a lot of BIP kind of in the ER. This should all be ER, and maybe some in Golgi. And then you can think of the red as your memory. So you see BIP in the ER and Golgi, and you decide to look for a retrieval signal. Do you see a retrieval signal? How do you prove that it is a retrieval signal? What experiments would you do? And what results you expect. So take a minute to talk to your neighbor and think about, look at the sequence. Is there an ER retrieval signal? That's what we were just talking about with KDEL. And most importantly, how would you test your hypothesis? Oh, yeah.
Oh, wait, no.
Okay, guys. We already know. We already have the Okay, guys. That was so much fun. We had so much fun. Who's still talking about Cadell and who's talking about the Ravens game? Who wants to take my son to a Ravens game? He's dying to go, and I really would rather be tortured than have to sit with the ball. All right. Good. He's 4. He's like, the Ravens, how are you? Take me to a Ravens game. I'm like, you're expensive. I pay a lot of money and hate my life, right? I think I'm going to take him to a Tigers game. Right? That seems like a better choice. Okay. So, the question was, do you see a retrieval signal? I'm guessing you all do. If you all saw Cadell. I did get a question of how is that different from a signal sequence? Who said it's not? Well, it's not conceptually in the sense that it's somehow going to bring the protein to the ER. But it's totally different in the sense of the signal sequence is about routing proteins to the secretory pathway, and Cadell is about retrieving proteins from the Golgi that are already in the secretory pathway from the Golgi to the ER. The signal sequence is N-terminus. It's the first little bit it's a great, you gave me such a great idea for an exam question. We'll write it down quickly. N-terminus, first little bit, that's translated, that SRP binds, so it brings the whole thing into the secretory pathway. This is C-terminus, right? Okay. So you see a retrieval signal, fantastic. How do you prove that this is, in fact, a retrieval signal? So someone said sequence it. Well, I'm like, well, you have the sequence. So you have the sequence already. You see Cadell. What do? Yes?
Could you remove the Cadell sequence and see does it go back to ER without it?
That's the perfect experiment. Can you just remove Cadell? So anytime you think something does something, get rid of it and see if it doesn't do that thing anymore. That's pretty much science. That's all of science right there. Actually, cell biology, molecular biology. Hey, I think this does this. Let's mutate it and see if it stops doing this. So you don't need to be like dual pulse phase. I mean, you can get as detailed as you want, but sometimes people don't have that experience in the lab. I'm not looking for detailed laboratory experiments. I'm looking for conceptual things. Mutate it, cut it off, see where it goes. The other thing you could do is maybe cut off the last little bit of this protein and add it to a protein that's not an ER resident protein and see if you can send it to the ER. Any other thoughts that people had? Who feels like they totally get it, they wish they understood it, but they're on their way, we're going to be okay? Who wants to start crying? Okay, good. No tears, no tears. Okay, so what results you expect, right? So if you cut it off, it shouldn't go it shouldn't go back to the ER, but where would it go? Out of the cell. It would be secreted, right? Because the default pathway, unless diverted, is secretion, yes.
So in that case, would you see the stain outside the cell?
Yeah. Yeah. So if it was a live cell imaging assay. Yeah. So you would see the staining outside. So in this picture, these are fixed cells. So it's like a snapshot in time, so you actually wouldn't see it. But what you would see is a lack of stain. But if you did live cell imaging with the fluorescence, it's really hard to do. I guess if you had GFP tag protein, you can watch it, leave the cell, I don't know. But yeah, it should be out. The other thing you can do is, let's say you want to do Western, right? You can look for the presence of a protein inside the cell in the ER, because you can actually isolate your ER, see if it's in the ER, or you can see it's in the supernate in your cells that are growing. Okay, good job, guys. The Golgi, oh my god, are we just getting to the Golgi? I've been slow today. I'm really glad I got rid of those first 4 lectures of the semester. Okay. Did you guys realize I got rid of 4 lectures in the beginning of the semester? Yeah. Okay. So Golgi, right? So proteins enter the cis-Golgi network from the ER, and they exit the trans-Golgi network. It's just cis and trans. It is what it is. The proteins exit from the trans-Golgi network to lysosome, plasma membrane, or secretory vesicles. This is just showing what Golgi looks like, and then these pictures here are showing your nuclear, so you can see your nuclear envelope up at the top, and ER, rough ER, because you see little dots of ribosomes there. There's vesicular tubular clusters here moving towards the Golgi. And your Golgi, sometimes people call the Golgi stacks because it looks like a stack of pancakes. Who's hungry? Now, pancakes. Yeah, but at least I'm teaching at like 3. I was always hungry then. Okay, so there are different. So another thing to think about when you're thinking of organelles and cells. Remember, cells aren't all the same. They're different types of cells that do different things, and they're going to have different amounts of different types of organelles. So, for example, cells, goblet cells that are specialized for secretion, goblet cells in the intestine, they secrete mucus, which is basically a mix of glycoproteins and proteoglycans. So when I say glycoprotein, you should think glycoprotein. Glyco has sugar. It's a protein that's glycosylated and secreted. Oh, it must be in the secretory pathway. Oh, secreted, secretory pathway. Making all these connections. Yeah. So these cells that secrete mucus have a lot of Golgi. What cells have lots of mitochondria? Muscle, yeah, muscle cells. Okay, so what happens in the Golgi? Basically, those end-linked oligosaccharides that were added in the ER and were trimmed a little bit in the ER and involved in protein folding and all that stuff, they are trimmed and changed, modified in the Golgi. Right, so in the ER, you've got, so you've got your original oligosaccharide that's added to your protein, and then these different enzymes are going to trim so it looks like this. Then you're going to go to the Golgi, and then there are different enzymes there that are going to modify those sugars further. And this is useful if you're trying to study where your protein is, because you can actually use an endoglycosylase that specifically recognizes high mannose sugars and examine whether or not your protein is in the ER or earlier in the Golgi or not. Yes?
You said that proteins enter the cis-Golgi and then trans-Golgi. That's only the secretory pathway, right?
Yeah, so all of this is secretory pathway. Yeah. Yeah, so not all proteins enter the secretory pathway. So then they're never going to be in the Golgi.
But if they are on the retrograde pathway, they would enter the trans and then
Okay, so you're connecting it to recitation. That's great. Right, so proteins are going the retrograde pathway, as we're talking about in recitation, which is rare. So that's one thing I was saying to people on Tuesday, is retrograde pathway is being used by toxin. It's not a normal flow of proteins. Your normal flow of proteins with endocytosis is to the lysosome. But yes, some proteins do follow this retrograde pathway, and you're right, they're going to go trans and cis. Yeah. Yes?
what about the ER resident?
And then, yeah, right. So nothing in science is a rule. I guess something is. There are those laws of thermodynamics and whatnot. But But yeah, so there's always an exception. The resident proteins are going to go Golgi, cis, ER. Yes. Yes. Okay. So trimming and modification of sugars. That's what happens in the Golgi. Right? So you have trimming of your N-linked oligosaccharides that were added in the ER. You get O-linked glycosylation. You get proteoglycans added. You get sulfation. Who is going sulfation? That was in the paper. Is there one person in this class that's going sulfation? That was in the paper. Remember the paper, this paper for recitation for this week? They were looking at the different aspects of the pathway, and they did this experiment where they were looking at, what was it? Right, retro 1 and 2, but they were doing this assay where they were looking to see if the protein made it to the Golgi, because there was a protein that could be modified and sulfate. They're looking at sulfation. Sorry, sulfation happened to the Golgi. Just when you look at that paper, go, ooh, sulfation, we learned about that. Addition and modification of sugars. That's the thing. So why do we care? Why are there glycosylated proteins? Why? What's the purpose? Yep. Okay, well, so one thing you're thinking of is adding mannose-6-phosphate. That is one thing that happens to proteins that go to the lysosome. And yes, it does mark lysosomal proteins destined for the lysosome to go there. So that is one thing, but not all proteins get mannose-6-phosphate, but all proteins that come out of the Golgi are going to be glycosylated. Yes?
They are
Helping with protein folding. Yep. Yes.
Isn't it something like
Okay, involved in signaling, right? Because remember, a lot of these proteins are going to be receptors that live on the cell surface and are highly glycosylated. And that is involved with signaling. Yep. Anything else? Well, I had a list. So what did I have? So the first thing to think about is, look at all these enzymes that are involved in trimming and addition of sugars in the Golgi. So if there's so many enzymes that do this, it's got to be important because your genome doesn't just keep things that have no use, right? Right? We have a greedy genome or something like that? No, we don't. So they must be important. So yes, protein folding. Also, they protrude from proteins that are on the cell surface, and so that's actually a form of protection. So you're limiting interaction with other proteins, such as proteases. Cell adhesion, which I've got a slide on coming up, and then it's involved in regulation of cell signaling also. So one example with adhesion, what does adhesion mean? Sticking to, yeah, adhesive tape, right? So adhesion, when do you need adhesion when we're talking about cell biology?
To keep things on the surface.
Hmm?
To keep things from falling off the surface.
Keep things from falling off the surface. Now think about adhesion of the cell to something. Okay, so when you're having fusion of two membranes, now let's think outside the cell. We're thinking cell, so fusion of membranes, we're talking about secretory pathway vesicles fusing. Hopefully we're not having cell-cell fusion, because that's not always that good. Yep. When you have to phagocytose something, okay. What else?
Clotting.
Clotting. Clotting factors, I wasn't thinking about that. But yeah, maybe, I think. I'm not sure. Is that right? Well, I know you have your clotting factors. I'm not a physiology person. Who wants to come up here and go through that pathway? No? I feel like you're right, though. Who else? Yep. Huh? receptor? So receptors, right? So that's what I'm showing here actually, right? So there are certain receptors. Selectins are one kind of receptor. And they have a lectin domain. Lectin, that just means that this part of this receptor binds to sugar. Lectin domains bind to sugars. And in the immune response, so forget the immune response for a minute. Where are your white blood cells normally? Well, they're everywhere normally, but where's the bulk? If you had to get white blood cells, where would you go? What part of your body? Did I hear someone say blood? No? Lymph. But lymph, I mean, that's intermingled with your blood through your circulation, right? So you get white blood cells out of your blood, right? That's what they do when you go to the doctor and they take your blood and they count your white blood cells, see if you have an infection, all that stuff. So anyway, so if you have an infection somewhere, the white blood cells need to get out of your blood and into the tissue where the infection is, right? You guys believe me? No, white blood cells never go to sites of infection. Never. Yeah, so that's what's happening here, right? So this is your blood vessel, and these are your endothelial cells that line your blood vessels. And so those cells need to come out of circulation and go into the tissue somehow. And so what they do is they squeeze through those junctions between your endothelial cells. How do they do that? Well, one thing they do is they will interact with these receptors, these selectins. So you'll have, well, wait a minute. Yeah. So the white blood cells have selectins on them, and they will bind to glycoproteins. Right? Let's go back to where we're coming from, right? So glycoproteins, glycosylation of proteins, Golgi, right? Glycoproteins, they're on your endothelial cells. And so your selectins it's kind of like Velcro. Your selectins on your white blood cells are going to bind to the glycoproteins on your endothelial cells. And it's going to be like Velcro. It's going to stop the cell from moving. Because remember, you've got this blood that's rushing by. These cells need to slow down and stop so they can squeeze through your cells in your blood vessels to get into tissues. So that's one example, just one, of how glycoproteins work, or a role for glycoproteins, right? So selectins, they bind to glycoproteins on vascular endothelium, and they're involved in movement of immune cells from blood vessels to lymph nodes and into tissues during infection. Okay, so I just said there's all these different modifications that are happening. There are all these different enzymes in different compartments of the Golgi. And yeah, in the different compartments, different process, different trimming or addition of sugars takes place because different enzymes live there. How do we know that? People have actually stained for different enzymes, and they localized to different compartments. So if you look at, this is unstained, if you look at your Golgi and you stain for different types of enzymes, you'll see that some are in the cis, some are in the trans, and so those different modifications happen as the proteins are moving through the Golgi. So I just said proteins moving through the Golgi. Well, so that means that I'm a proponent of the vesicular transport model, apparently, because I always think of things that way. But there are actually two models for transport through the Golgi. One is the one that seems to make the most sense, which is that you have your vesicular, your vesicles are going to fuse with each compartment of the Golgi, so your protein will go into that compartment, be modified, bud out in a vesicle, go to the next compartment, right? This is this vesicular transport model. So just kind of hopping through the different compartments using that vesicular transport. But then there's this other model, which is histernal maturation, which really is more talking about how the enzymes are kind of moving through the different compartments. Just acknowledge the fact that there are two models. One where the proteins are flowing, and the other where the enzymes that do the modifying is kind of, they're moving. And that's that for that. Okay, I think we're gonna stop right there, if that's the right time. Sure.